Firstly, great to be able to talk to you all today and share something a little bit about Gordian Pharma with you. My name's Mike Hudson, and I'm running the program, been involved in it with several years, and want to talk to you about it today. It's a really interesting molecule targeting the chemotherapy arena and particularly relevant in pediatrics because of its functional properties. And the problem is that cancer is very variable, and it's also still the leading killer of disease-related deaths in children. But it's not just about the ones that don't make it. Even the ones that do make it, the survivors, nearly a third of them suffer very complex and often life-threatening, certainly debilitating side effects. And when we look in the $170 billion of drugs used to treat cancer every year, what we find is that, a big bunch of them have side effects that challenge their use in children. More modern, newer drugs that are pathway targeted are often unsuitable for use with children. So those great drugs that are progressing now in adults may be inappropriate again for use in children. And you can forget about people in low or middle income countries being able to afford those drugs for sure. So there's an urgent need for product innovation. And when we look into the heterogeneity of cancer, what stands out really quickly is that glioblastoma, one of the more aggressive forms of brain tumors, is the real one that should be the target. It has a very, very poor 5-year survival rate, less than 10%, and that draws attention to it as still one of those killer diseases. And it's relatively rare in that it's a cancer that occurs both in adults and children. And obviously the additional incentive of a cure or a treatment that would work in both populations, adults and children, provides a massive financial incentive to help this program run. So if we look at what is current practice, most drugs can't penetrate the brain, and therefore the only treatment initiated is around surgery. And then there is the option of following that or combining it with radiotherapy or some local drug treatment. And the real problem is that even after removal of the tumor, there is a very frequent, high frequency of recurrence. And the recurrence is always within a couple of centimeters of the original site, and it's visible within 3 months. So there was a very good strategy of applying local chemotherapy to try and disinfect or clean up after the surgical procedure and kill any tumor cells or dormant tumor cells that might have been missed, because these are very invasive and the borders are hard to define. But the problem of using some small wafers with a slow release of a chemical chemotherapy agent is that firstly, those rigid wafers didn't stay in place. So they didn't provide uniform protection into the cavity after surgery. And the drug that was used and is currently licensed is a mustard gas derivative. And it is a very noxious and pretty horrible chemical that's been around a very long time. And that is why the median survival today still in glioblastoma is around 15 months. So, this is a real problem. But the strategy was good. And so let's look back at that and let's think about carmustine and look at MTL-004, which is the molecule that we've been working with now for a number of years. And MTL-004 has familiarity in that it's a drug that works by damaging the DNA, like many of the chemotherapy agents, which got two very unique features. The first is that it's deactivated on contact with blood. Now that actually makes it almost impossible to use in the same way as other systemic chemotherapy agents that are injected. But it is intrinsically, therefore, very safe because it stays exactly where it's applied to the tissue. And its effects are only manifest at that local tumor site. The second dimension is that it is incredibly toxic. And I mean orders of magnitude, 100,000 times more toxic, because it forms very deep bonds within the DNA structures that stop the strands separating. And nature hasn't developed ways to repair these particular types of damage to DNA, unlike the damage that occurs by many of the other drugs. So if we compare the amount of MTL-004 that's bound to DNA with other cytotoxic drugs, we see that 70% of our drug is in these very resistant, long-lasting, toxic bonds, and that is really critical, because it means there are no survivors that are simply mutated or damaged and can lead to secondary cancers. And that's a massive risk associated with these other chemotherapy agents, where what doesn't cure you can ultimately kill you. It also means that it lasts and provides a period of protection that is 5 to 10 times longer than any of the other existing chemotherapy drugs. So it's a very interesting molecule. So let's just think, from current practice, let's forget about putting those wafers into the cavity after where the tumor has been removed. Let's look at using MTL-004, because it's much more potent and safer than carmustine or many of the current chemotherapy agents. And then applying that using a gel like toothpaste, going back to our first presenter today, but this is a gel that's used to fill the cavity and is then naturally resorbed. So it gives very uniform contact and exposure and protection. So we took this information about the functionalities and the properties of MTL-004 to the European Medicines Agency last November, and explored with them using ICHS9, which is an abridged pathway for products that have potential to treat advanced cancers where the prognosis is very poor. And they have agreed and confirmed to us in March this year that glioblastoma, advanced small cell lung cancer, and invasive bladder cancers would all be eligible targets for the direct treatment using MTL-004, and that this pilot would benefit at least 350,000 patients, even in these niche defined areas of cancer. And from an experimental perspective, it would also allow us to compare the binding and cytotoxicity of the drug and its safety with 3 of the biggest selling and most widely used chemotherapy agents today: carmustine in the glioblastoma, in the brain tumor, cisplatin in lung, and mitomycin in the bladder. So our lead program in the glioblastoma in adults and children was approved as being one of the priority programs for fast track evaluation. The team is a very small team. It's led by myself. I'm a first generation biotechnologist from the mid-70s, worked with a number of companies in orphan drugs, pediatric drug development, from IP spin-outs from universities through to listing of public companies. I'm joined by Richard Knox, who actually discovered MTL-004 and has been the real inspiration behind the program, spent all of his career developing and validating this compound. And then we're kept on the straight and narrow by Judith and Mike, who are colleagues who manage our IP, our finances, and provide overall chairmanship. The real resource that's now moving this program forward very quickly, and as our audience is really the regulators, is a group of specialist CROs. They, between them, employ about 1,000 R&D staff, and they each of them have a very particular reason for being at the table with us. Arkinova, formerly part of Sanofi, marketed the second biggest platinum drug, so a lot of expertise in monitoring chemotherapy in body fluids. Ligand Pharma supply the majority of the top 20 pharmaceutical companies with complex excipients to protect and deliver the active pharmaceuticals in formulations. Vectura is a company developing the drug product and also does inhaled delivery, which is very important for our lung validation. And then OncoDesign is a public company in Europe that has been working and has their own oncology program, but selectively works with others with non-competing programs. But they have some very advanced molecular biology resources in the oncology space. And then finally, we've got really 2 working relationships currently, but this list will grow in terms of clinical and patient engagement. The Brain Tumor Charity have been continual sponsors, supporters, encouragers of this program, obviously, but they've also provided some great network contacts for the preclinical validation studies, as well as moving this closer to the clinic. And ultimately, we do need to work with the WHO list of approved medicines to be able to make these products accessible around the world. we've got 3 objectives now with the current fundraising, which is a $3.5 million raise. We had a seed raise last year. That is to complete now all of the preclinical work that the European Medicines Agency have agreed with us, to consult with the EMA and FDA on this next occasion during the course of this year, to define exactly the clinical work that we will be required or allowed to conduct, and to file both our orphan drug and pediatric applications. And really good news for us, 10 days ago, the FDA and EMA published a joint paper to specifically address pediatric cancer medicines jointly, which has been so long overdue, I have to say, but that's a great assistance that we're able to play and work within the regulatory framework as a key program. By this time next year, or in the 3rd quarter, we should be able to initiate several clinical programs to take this through to completion. So really what we're looking for today at this point in time is to be able to complete this program of work. It's about $3.5 million. In the last few weeks, we've already secured at least half of that, in fact, over half of that, almost 60% as of yesterday. So we are looking for about $1.5 million now to complete this round, but also to bring some US stakeholders to the table with us who can share and be part of the journey at the next stage. That's really key for us. Be very happy to take questions, and just like to thank the Centre for Advancing Innovation and all of you today for listening and encouraging us through this journey. It's been wonderful experience to be part of the program, and I look forward to the next stage.
Thank you, Mike. I think we have time for one question. So Dr. Hudson, at cellular level, does MTL-004 cross-link with DNA similar to MMC, cisplatin, and other ICL inducers?
Well, all of those molecules form damage to the DNA and link with the DNA at different points. Sometimes it's external and it's a single deletion or damage, and those are the ones that may be repaired but not always correctly repaired, and so have a potential risk associated with them for mutagenicity. The ones that are able to get inside and form interstrand cross-links are much less repairable. And we find that our molecule has a much higher capacity and potency to form those interstrand cytotoxic links than anything else. So literally almost an order of magnitude greater. But the principles of damaging the DNA are shared between all of those molecules.
Thank you very much, Mike.